Welcome from Dresden. I'm Gene Myers. Together with Eric Jarvis, Richard Durbin, and Adam Philippi, my team here has been involved in sequencing and developing assembly algorithms for the groundbreaking vertebrate genome project since its inception. Today, bioinformaticians Tom Brown and Martin Pippel are going to describe how we're organized here, our technology base, and what we've been up to lately.
Here at the Dresden Concept Genome Center, we're a collaborative effort between the MPI-CBG and the CMCB at the TU Dresden. We combine forces to offer both short and long read sequencing, but here at the MPI-CBG we mostly focus on long read sequencing with the aim of assembling genomes from a variety of species across the globe. We mostly focus on sequencing bats, covering all bats across the world, fish from across Europe, and even up to some endangered turtle species. We follow through a full sequencing and assembly pipeline from receiving tissue in the lab, extracting and sequencing DNA, through to getting the data onto a compute cluster, assembling a genome, and then ultimately releasing this into the public domain. We aim to create high-quality reference-level genome assemblies, and to us, with our current sequencing capabilities, this means satisfying the VGP's 2020 metric of a conjugate N50 of at least 10 megabase, 95% of our genome scaffolded into chromosomes, and a QV50 value, or at most one error per 100,000 bases. At the Genome Center, we perform all types of long-read sequencing required to produce high-quality genome assemblies. We primarily rely on PacBio's HiFi sequencing, which has an average error rate of only 0.1%. We also perform long and ultra-long sequencing on Oxford Nanofor's PromethION platform, useful for resolving highly repetitive regions which HiFi or Illumina sequencing cannot resolve. For scaffolding, we use long-range information collected from Hi-C sequencing and bio-nano optical mapping.
We, at the MPI-CBG and the Center for Systems Biology Dresden, where we are now, are the main sequencing and assembly hub for the BAT1K initiative, which aims to produce high-quality genomes of approximately all 1,400 bat species around the globe. Currently, we have sequenced 33 bat genomes across 15 bat families. Over the next 2 years, we will support more institutes to set up their assembly pipelines. The development of the highly accurate long reads that are produced from the PacBio HiFi sequencing platform has led to a dramatic increase of the production rate of the genomes that are produced by the Dresden Concept Genome Center. Currently being established as part of the Vertebrate Genome Project, the European Reference Genome Atlas, or ERGA, which aims to sequence and assemble a range of threatened, endangered, or key biodiversity European species and releases them to the public domain. The Sequencing Center is heavily involved in the pilot phase of this project, providing sequencing and assemblies for a number of species, including the endangered Spanish minnow carp. On top of this, we are providing protocols, pipelines, and our expertise with all other sequencing and assembly centers around Europe. This will contribute to the ambitious goal to sequence all life in Europe.